Hello students, welcome to week 4 of Biology 11. So today we're going to be covering enzymes. So what is an enzyme? An enzyme is a substance that helps to speed along chemical reactions. What exactly are chemical reactions? So in chemistry, we denote reactions in this format using A plus B with an arrow pointing to C plus D. In this formula, the A and the B represent the reactants of a chemical reaction, meaning the molecules that are undergoing a change, and C and D represent the products, which are the final molecules at the end of the reaction. So in biology, a cell is using many chemical reactions to support life. Most importantly, for example, is converting food to energy. So, when we eat our breakfast, our body will break it down into the simple substances, the macromolecules, that we can use. And this is important. This is a lot of different chemical reactions going on to actually get this process done. So what is an enzyme? Exactly. It's an organic catalyst that speeds along these chemical reactions. So what exactly does this mean? Many chemical reactions will occur naturally if you just put two substances that want to react together based on their chemistry. They will react, but it depends on many factors, how efficient this process is. So this can depend on the amount of substrate or product in a system or solution. It could depend on the temperature, and it can depend on the amount of available energy, and all these things will influence how the speed of the reaction varies. So in general, living cells are pretty lazy. They want to do things with the least amount of energy involved as possible. So enter the enzyme. An enzyme actually lowers the amount of energy needed to perform this chemical reaction, meaning it just makes the job easier. So if you look at this graph here, you can see that the amount of energy required to accomplish a specific reaction, as depicted by the blue line, is higher than the amount of energy needed to complete this reaction with the presence of the enzyme, which is depicted as the red line. So basically, it's just saving energy. It's making the job of the cell a lot easier. So enzymes are specific only to one type of reaction, meaning they only catalyze one reaction. They have one job. And this is important in terms of regulating these reactions, because we don't want chemical reactions just going on in a cell. That would be chaotic. In an enzymatic reaction, we use a specific term to refer to the reactants, calling this a substrate. So an enzyme will act on this substrate. So in today's lab, we're going to talk a little bit about catalase. So how does an enzyme accomplish this? So there's a little bit of chemical geometry involved in this. So the area on an enzyme where this chemical reaction takes place is known as the active site. So you can see this depicted in this diagram. Prior to binding to a substrate, that active site of an enzyme assumes a certain shape. So just think of puzzle pieces. So as the substrate encounters the enzyme and binds, the enzyme itself undergoes something called a conformational change, meaning a change in the shape of the enzyme. The enzyme and the product then form this complex. They bind to each other while the chemical reaction is taking place. So in this example on this slide, this is an enzyme breaking down a substrate into two products. Once the reaction is finished, the product can leave the active site of the enzyme, and then the enzyme returns to its original shape, its original conformational shape. So this means that we can recycle enzymes. They're not changed permanently by this process. They're not actually undergoing a chemical reaction. They're facilitating a chemical reaction. So once the product leaves the active site, the enzyme can then bind to another substrate and repeat this process. So enzymatic reactions are depicted using our formula as E plus S, so enzyme plus the substrate, an arrow, and then they form ES, which you have to think of as the enzyme substrate complex. And then you add another arrow, and then you get the enzyme and the product. So the enzyme is remaining the same throughout this entire process. So there are two basic types of enzymatic reactions. The last slide was showing you an example of an enzymatic reaction where the enzyme is breaking down a substrate into two products, and this is known as a degradation reaction. Synthesis is the opposite of this process, in which two substrates are combined to form a larger product. And most enzymatic reactions in our body can pretty much be categorized into these two types of reactions. So catalase. This is the enzyme that we discussed in this lab. So this is an enzyme that facilitates the breakdown of hydrogen peroxide, and it's located in an organelle known as the peroxisome. So this is a really important enzyme because hydrogen peroxide is actually a byproduct of many chemical reactions in the cell, meaning it's just left over from all the reactions that a cell needs to undergo in order to survive. But hydrogen peroxide is a harmful waste product. In particular, it's interesting to me because I study genome damage, and hydrogen peroxide attacks DNA and damages DNA, which you can imagine is not good. It also can attack lipids, which, as we also learned from last week, that lipids are an important component of the plasma membrane. So this can, this can cause serious problems in a cell. And yeah, it really trashes chemicals in a cell. So this is also why hydrogen peroxide is a good antiseptic, which is, you might know it just from having this in your first aid kit. It's just the bubbly stuff that you pour on a cut. And so this is why it's a good antiseptic, because it will kill cells this way. But we need to manage this in a cell in order to survive. Otherwise, the cell is just going to get trashed and die. So as I mentioned, because of the conformational shape, many different things can affect the activity of the enzyme, and this is actually the purpose of the lab exercise in our manual. So the temperature is a very important condition that can affect the activity of an enzyme. So this can change the shape of that active site on the enzyme. This is a process known as denaturation. So the best way to understand denaturation, if you think about a scrambled egg, which is in a liquid state, and then you heat it, it puffs up into basically a different shape. So this is what's going on when you heat up any sort of protein, which is what an enzyme is. It's a folded up protein. So you add heat, and it loses its structure, and it just becomes scrambled eggs. And this obviously is going to affect how often that a substrate can contact that active site and whether it can fit into that active site. Another important condition that affects enzymatic activity is concentration. So how often is an enzyme coming into contact with the substrate? If there's a ton of enzyme and a ton of substrate, you're going to get a lot of reactions. But if you have only a little bit of enzyme and a ton of substrate, it's only going to progress based on the availability of the enzyme. So remember, the enzyme can be recycled, so it can do a job and then release its products and bind to other substrates, but it's going to be busy doing all of these chemical reactions, and that's going to affect the rate of a reaction. pH can also affect the conformation of the enzyme and the active site, so this can also affect the rate of an enzymatic reaction. So this is important in the digestive tract. I think I alluded to this last week, that the pH of our GI tract changes drastically as food travels from our mouth to our anus. And this is because all of the enzymes that are used to break down different macromolecules, so that's carbohydrates, proteins, and fats, occur at different pH. And so these enzymes function at different pH, and so they can only function when the gastrointestinal tract is a certain pH. So this is very important, and we'll talk more about this when we discuss the digestive system. So I was planning to do some kitchen science experiments, but the week kind of ran away from me, so I'm going to post some videos of other professors who have made videos of the potato catalase experiment. It's actually a very interesting and very simple experiment that you could do on your own if you so choose. I can post a protocol, or you can just follow a little bit of what we do in the lab. The potato catalase is essentially just potato pulp. It's just chopped up potatoes and water. And this releases the catalase. And then you can use regular hydrogen peroxide from your first aid kit to test what exactly happens. So I'll post a video for you on Blackboard so you can look at it. And then you can complete the manual assignments, which I will also post along with this video. All right, thanks for listening, and I will see you next week.